Gilteritinib is a selective and potent FLT3 inhibitor. It's a type 1 inhibitor, so it's active both against FLT3 ITD mutations and also the tyrosine kinase domain, or TKD mutations, that are both common in AML. And the FLT3 ITD mutations have been really problematic because they're associated with high rates of relapse. But if either are present at relapse, patients have really dismal outcome, very short survivals, and poor responses to salvage chemotherapy. So this has kind of been a low-hanging fruit for drug development in terms of developing tyrosine kinase inhibitors, but to date, none of them has really stuck in terms of having a long-term outcome in patients with relapsed refractory disease because of a number of limitations. Either they weren't potent enough, or they had toxicity issues, or they led to resistance in the FLT3 receptor itself. So we would see patients with FLT3 ITD mutations who would respond to a drug and then lose response by developing a TKD mutation, and that's where Gilteritinib was developed, to kind of cover all of those bases, to be a well-tolerated drug with activity against all of the mutations. And on the ADMIRAL study, we put it head-to-head against standard salvage chemotherapy in the population of patients with either refractory AML with FLT3 mutation or relapsed disease after a prior CR. So this was a phase 3 study, 371 patients enrolled to it, and they were randomized 2-to-1 to either single-agent Gilteritinib or investigator's choice of standard chemotherapy, either high-intensity chemotherapy, reinduction therapy, MEC or FLAG-IDA, or low-dose cytarabine or azacitidine as the low-intensity options, which were chosen prior to randomization. The primary endpoints of the study actually were a co-primary endpoint of response in the Gilteritinib arm, which really was just used for regulatory submission. But the primary endpoint of the study was overall survival. And the study actually showed not only higher response rates in the Gilteritinib arm, but also improved survival in that group as well. The median survival for the chemotherapy group was 5.6 months, and it was 9.3 months in the Gilteritinib arm. And this is notable because it's a molecularly targeted agent. For the first time in the relapsed refractory setting, we're actually showing that picking a mutation and then targeting it leads to better outcomes. And so I think that's noteworthy, that really FLT3 inhibitors are the only drugs for which we've shown this kind of benefit in terms of using a targeted drug and showing an overall survival benefit in the phase 3 setting. The drug was also submitted for regulatory approval, as I mentioned, and was approved in the US and also in Japan in the past year. The main thing we see are cytopenias, which has been true on a number of drugs targeting FLT3. It's a hematopoietic cytokine receptor, so it regulates hematopoiesis and how many blood cells are made daily, so that's not entirely surprising. The other things we can see are largely asymptomatic laboratory value abnormalities. We can see LFT abnormalities with some regularity, and those were seen actually a little bit more frequently in the Gilteritinib arm than the chemotherapy arm. But overall, if you looked at all the toxicities, there are fewer serious toxicities in Gilteritinib arm when corrected for drug exposure, because patients took Gilteritinib much longer than chemotherapy, largely because they responded to it more frequently and then just stayed on study until either intolerance or progression. There were a few toxicities we need to monitor for in the drug, and in its approval, there's recommendations for monitoring of QT prolongation, which was not very common but did occur in some patients. There's also some risk of CK elevation, some risk of differentiation syndrome, and a few patients had elevations of lipase, with relatively few patients who had pancreatitis. There's also language regarding PRES, or posterior reversible encephalopathy. Again, very rare, but if it happens, it's serious. So the message is there's a new standard of care for relapsed refractory patients with FLT3 mutation, which means we have to find them. So the thing that's notable about FLT3 is it's often retained at relapse or in patients who are refractory to induction, but there are times when that actually changes. The patients didn't have a FLT3 mutation when initially diagnosed, and then it shows up at relapse. And clinically, the way you spot that is when patients have a rapidly rising white blood cell count. That's when we tend to see these mutations. And they can be lost also, so it's worth retesting at relapse because if you look at the response rate of the drug in phase 1/2 testing, where we did not require patients to have a FLT3 mutation, it was really quite low in patients who didn't have FLT3 mutation. So there really is only a benefit in the mutated patients.